This next segment is a little unique. It looks at the use of antibodies as drugs. Okay, so why would you use an antibody as a drug target? Well, you kind of have to go back to some of the physiologic outcomes to what an antibody does upon binding to a cell. This is taken from a nice review in Nature, cited here. And then this is looking at, obviously, a specific antibody against a B cell. For the sake of this lecture, you can just ignore that component. We'll talk about B cell specific antibodies in another disease state. This can easily be a specific CD marker for a T cell, but the concept is the same. And so there's three main mechanisms of action that antibodies can produce to actually act as a drug. Okay, and so they can induce, and you remember this probably from last year, they can induce antibody-dependent cell cytotoxicity. And so this involves the use of some of these innate immune cells as well as the FcR region. And basically when these cells are kind of bound, or when this antibody is bound, these cells bind to that antibody and they mark the target for death. The other mechanism to be aware of is the complement-dependent cytotoxicity mechanism, where essentially the binding of the antibody to the target cell actually induces the complement cascade, of which the membrane attack complex is kind of paramount. And then again, that marks the cell, the target cell, for death. And then of course there's direct apoptosis, where the antibody that you're supplying could be labeled with some type of radioligand or some type of toxin that gets internalized at the receptor level, and then again marks that target cell for death and destruction. Okay, and so one thing I want to point out is that when we talk about antibodies being used as drugs, we're not talking about endogenous antibodies that are already within the patient. We're talking about bioengineered, genetically modified antibodies that are grown in a laboratory and are actually specifically targeted to a specific receptor that plays a role in the disease pathogenesis. And we'll go through some of those in the next coming slides. Okay, so here's kind of a review of all of those different mechanisms. A naked monoclonal antibody, so MAB is the pharmacologic and immunologic acronym abbreviation for a monoclonal antibody. Monoclonal antibodies that are supplied as drugs typically induce cell death of the target cell through either of those two mechanisms, antibody-dependent or complement-dependent cytotoxicity. In this particular diagram, the tumor cell is indicated, and so that's where a lot of these monoclonal antibodies were first kind of developed, was they would actually target tumor cells for destruction. Later, as we've kind of gone through some of these various studies, we found that we can actually apply that same logic to specific cells in the host, in the human patient, that are kind of driving pathology. And so in this particular lecture series, we were talking about so many of these immune cells, particularly T cells, that were driving some of the pathogenic changes in the GI system. So these are, overreactive T cells that are contributing to Crohn's disease, possibly some of these other pro-inflammatory type of GI diseases. Okay, and so you can put radioligands on these. You can put all kinds of molecules on them, and the concept here is the same. You basically mark this particular target cell for death and destruction by the body's own kind of surveillance mechanisms, using these drugs to kind of show the particular patterns of immune cells that need to be able to target this particular cell. So just to kind of summarize, the mechanism of action of monoclonal antibodies include antibody-dependent cell toxicity, complement-dependent cell toxicity, and then you can use the conjugated toxin to basically cause this monoclonal antibody to be internalized, and then that will cause apoptosis. So any of these mechanisms will occur. So what do these drugs look like? Well, there's two targeted pathways for some of these GI inflammatory diseases. One group of drugs target the TNF alpha pathway, so the tumor necrosis factor alpha pathway. This is a pro-inflammatory cytokine that really plays key roles in leukocyte movement and really has a definitive role in Crohn's and ulcerative colitis and celiac and some of these other types of diseases. The T cell activation pathway you can see in many different lights in terms of how these T cells really drive disease progression. If any of you are even remotely interested in hematology or oncology, this particular paper that is linked right here is, I would say, probably a must read. I've probably read this paper 4 or 5 times. And it's called The Paradoxical Roles of the Immune System During Cancer Initiation and Progression. And it's really interesting. What it does is it paves the way for how normal immune system activity actually contributes to pathogenesis of tumors. And it really kind of provides a rationale for all of the drugs that we're going to talk about in this next video. Okay, so the first group of drugs are going to target tumor necrosis factor alpha. Why do we want to target this particular cytokine? Well, we said it's pro-inflammatory, and it really kind of directs the innate immune system to various pro-inflammatory type processes. This also plays key roles in the adaptive immune system, particularly among Th1 type cells. But they also have, TNF-alpha also has a role in the non-immune cells, fibroblasts, smooth muscle cells, all of which are a component of wound healing and inflammation. So TNF-alpha binds to the TNF receptor, and that activates what's called the NF-kappa B cascade, which is a very large signal transduction cascade, and the outcome of which is going to be the transcription, growth, and expansion of pro-inflammatory cytokines that really drive progression of a lot of these diseases. All right, so there's 2 groups: infliximab, which is given IV, and these 3, which are given subcutaneously. And essentially what you're doing is you're really inhibiting the cascade. And so it doesn't really matter at this point, but some of these drugs inhibit circulating tumor necrosis factor alpha. Some of these drugs actually inhibit the receptor at the target cell. The outcome is the same, though. There's going to be an attenuation of signaling of tumor necrosis factor alpha through the body, and that's the thing that's going to be driving pathogenesis. So if you halt the signal, you can control the pathogenesis of some of these pro-inflammatory diseases. Let's end there with the tumor necrosis factor pathway, and we'll pick up with the T cell pathway next.